All right, guys, so I'm obviously not here today, but I want to go over the 2.3 and 2.4 sections of your study guide so that you are prepared for your test tomorrow. Remember, your test tomorrow is over all of Unit 2, so make sure you're using all the resources that are available on Google Classroom where it says Unit 2 test. There are videos, Quizlets, quizzes. I have all kinds of resources. Make sure you're using them all so that you can do well. But looking at your review packet for Unit 2.3, the cell cycle and division, big things we talked about in 2.3. What's happening in a cell? So it's making proteins. Where's that happening? In the ribosomes. So what happens to the proteins after they're made? This is that pathway that we talked about in 2.3 where the proteins are going to be made by the ribosome, travel through the ER, organized, packaged, and shipped by the Golgi apparatus. And then they can be shipped out through the cell membrane for other cells to be used by that cell, whatever its job might be. The job of the endoplasmic reticulum and the Golgi apparatus, which is kind of mentioned in this pathway, the ER is transporting the proteins throughout the cell, and the Golgi apparatus is going to package and label them. So sometimes it takes a couple proteins together to be able to do a job, or we need to put a marker of some kind so that if it's floating through the body, another cell can say, oh, that's the one I needed, and grab onto it. Why would a cell need to divide? So we talked about this back in 2.1, but it comes up again here. If it gets too large to be able to communicate within the cell or too large to be efficient anymore. Remember that cells constantly also are going to be growing, repairing, and replacing themselves. We talked about with HeLa, only about 50 cell divisions can happen. So we need to constantly be replacing these old cells when they wear out. Three phases of the cell cycle. Remember, interphase is that first part, and then we go through the M phase or mitosis, and then we end with cytokinesis. So during those three parts of interphase, there's G1, where the cell is growing and making its proteins and being a cell and doing whatever its job is. The S phase, where we actually copy or replicate the DNA. And then the G2 part where we're preparing for division with the organelles and the cytoplasm, making sure we have everything in order. The two copies of DNA that are created during that S phase are called sister chromatids. And remember, they're being held together at the centromere. We want to make sure that those two identical copies or sister chromatids stay together. Then we go into the M phase or the actual mitosis. And remember, that's PMAT. So the first part is prophase, where the spindles are going to appear, the nucleus disappears, and the chromosomes become visible for the first time. Then we have metaphase, where the sister chromosomes are going to line up in the middle, so those centromeres attach to the spindles. And then there's anaphase, where the sister chromatids get pulled to opposite sides of the cell. The last part of the actual dividing of the nucleus is that we create two separate nuclei, two separate nuclear envelopes, each with one copy of the DNA, and that is what telophase is all about. How is cytokinesis different in plant and animal cells? Where the last part is where we're dividing the cytoplasm. In a plant cell, we have to build a cell plate from the inside out because the cell wall is too rigid to bend, so we have to build a wall. With animal cells, the cell membrane is flexible, so we're going to be able to pinch it in the middle and be able to squeeze it from the outside to the inside to break that cytoplasm. So when a cell is going through the cell cycle, we know that there are feedback points. At the end of G1 saying we've grown too big and need to divide. At the end of G2 saying, okay, we replicate the DNA and we have enough stuff here to divide. And then again during the M of metaphase, end of metaphase, we're going to check and make sure did all of those copies line up in the middle like they were supposed to. But if at any point the cell does not respond to those checkpoints and those feedback signals, then we can have uncontrolled growth, which is what we see with the formation of tumors and cancer. As you go into your review next, we're into the unit 2.4, which we just finished, just took the quiz on Tuesday, so you should be really good with this. Plus, a lot of the things we've talked about along the way in 2.1, 2.2, and now just in 2.3 talk about these cell parts and what they're doing. So what type of microscope do we use? Well, we used the compound light microscope to see single cells like a bacteria cell. To figure out the magnification, we were multiplying the eyepiece times the objective. So on our microscopes, it was 40, 100, and 400 times magnification total. If a protist appears to be swimming to the right, it's actually swimming to the left if we're looking through the microscope. Or if we look, it looks like something's on the right side, it's actually on the left side of your screen. If it looks like it's at the top, it's actually at the bottom of your microscope. Remember, everything is reversed. Plant and animal cells do have a nucleus, which makes them eukaryotic cells. They also have other cell parts that have specific jobs, which are called organelles. Prokaryotic cells do not have either of those. Function of the chloroplasts. We talked about the chloroplasts and mitochondria in 2.2 with photosynthesis and respiration. They come up again here in 2.4. The chloroplast is going to be making food or glucose from the sun through photosynthesis, or the mitochondria is going to be breaking down that food into ATP and usable forms. Prokaryotic organisms are only made up of one cell. If we have a group of cells that are doing a job in a multicellular organism, we call them a tissue. A group of tissues working together are going to be an organ. Most organisms on Earth are going to be those unicellular or single-celled organisms. So we spend all this time talking about our multicellular selves, plants, animals, but most of the things on Earth are just going to be single-celled bacteria or single-celled protists, mostly bacteria. Make sure that you know all of the different organelles that we talked about, in class, so that you are prepared for them. They're in your notes that we did. They're on the study guide for section 2.4. We also have included for you some quizzes for review, so make sure that you do those as well. So check in this post for other things that your specific teacher has assigned for you to complete today, to be ready for tomorrow. Those things that are listed in the assignment with this screencast are going to be graded parts of the review. So make sure that you're doing whatever your teacher has assigned for you. Have a great day. If you have any questions, make sure you're posting them in the Google Doc so that Mr. Sandvik and I can answer those while we are at our conference. Good luck, guys.